So a lot of different topics in Chapter 4. This one is just to introduce you to a concept, another chemical reaction concept called redox reactions or oxidation reduction reactions. We're going to see these again in Chapter 5 and Chapter 6. So photosynthesis and cellular respiration are both these kinds of reactions, oxidation reduction reactions. So we're just going to introduce it now, and then we'll start actually talking about energonic and exergonic again. Oxidation reduction or redox reactions are any reaction where there's a transfer of electrons between two molecules. One molecule is going to give up an electron, and the other molecule is going to absorb or pick up an electron. So oxidation is the process of losing an electron, and reduction is the process of gaining electrons. So they always go hand in hand, because when one is oxidized, something else has to be reduced. So do you remember anywhere this semester that we've talked about something giving up an electron and something else gaining an electron?
Yes.
All right, so where? Yeah, ionic bonds. Okay, so an ion is formed by a redox reaction. So we have, I have this here. So Na, like NaCl, Na plus Cl gives you Na plus Cl minus. The electron has been transferred from sodium to chlorine. So is sodium oxidized or reduced? Yeah, it's oxidized. It's lost electrons, and chlorine then would be reduced. So one way to remember the difference between oxidation and reduction, because these words will come up again, is the mnemonic oil rig. Oil stands for oxidation is loss, and rig, reduction is gain. So that might be a good way of remembering these things. So oxidation is loss, reduction is gain. Yes?
Or you could think that reduction is reducing the charge.
Yeah, but we are going to see in photosynthesis and respiration, they are redox reactions, but the end products don't change their charge because you're also going to be transferring protons. So sometimes that doesn't work. But that is where that word comes from. Reduction comes from the fact that the charge is reduced, at least by it gains electrons. Yes. And that's where this gets confusing sometimes, because you're thinking, oh, it's gaining stuff, so it must increase in charge. But just like in ions, when it gains something, it's gaining a negative charge, so its charge is going to be negative. It's going to be reduced. So oxidation is loss, reduction is gain. So we're going to see lots of redox reactions in progress in the next two chapters, and they're always going to involve one gaining and one losing. One of the things we're going to see in both photosynthesis and respiration is something called an electron transport chain. So when a molecule gives up an electron, it's not just giving up an electron, it's also giving up some energy. Those electrons often have energy in them. So the receiving molecule will be gaining energy. So the reduced molecule is going to be gaining energy. So an electron transport chain is a series of redox reactions where you're passing energy in the form of electrons from one thing to the next. So you're transporting electrons, and in the process you're transporting energy. So we're going to see some kind of donor molecule first give up an energized electron, and then that energized electron is going to be passed through a series of proteins. So at first this protein is reduced, it gains an electron, and then it's oxidized, it gives up the electron. This protein gains an electron, then it gives one up. This one gains an electron, then it gives one up. And every time that electron is transferred, a little bit of that energy is siphoned off by the protein in the electron transport chain to do work. And the work that's going to eventually be done is going to lead to the production of ATP, which we'll see what that is in a minute. So both photosynthesis and respiration use electron transport chains in order to basically harvest energy gradually from electrons. And we're going to see especially when we get to respiration, that there's a lot of energy in the electrons in food, for instance. And so siphoning it off gradually through a process like this allows us to harvest a lot of energy from a molecule of glucose. So keep this in mind. This will come back in the next two chapters. But first, we want to talk a little bit about where this energy ultimately ends up. We'll go back in the next two chapters and talk about how that energy actually gets to that molecule. Okay, so before I go to the next thing, I wanted to go back to this slide. As we talked about this last class, we talked about exergonic reactions. So reactions where your reactant has a higher free energy than your product. Those reactions are going to release energy. And in the example of a hydroelectric dam, as the water flows downhill from the upper chamber to the lower chamber, it could be used to turn turbines to generate electricity. Okay. So that's one way of generating electricity. So our power plants, there are hydroelectric power plants, not really in Florida because everything's so flat. It's really hard to use that as a form. We will use solar power plants. We'll also use wind. We'll also just burn fuel as a way of generating electricity. Those are all exergonic processes. They all release energy. We capture it as electricity. So most of you have some kind of mobile device, right? You have a phone, you have a laptop, you have a tablet or something like that. Why is it, and this is a very simple question, why is it that I can walk around and use this, I'm recording myself with it right now, and I don't have to be tapped into a power generating reaction like a power plant or a wind turbine or something like that? Right, I have a battery, right? That's pretty common, right? You have batteries and things. So if I didn't have a battery in this device, I would have to have it either plugged in directly to that power plant, or I would have to have a little engine attached to it that I would start up every time I wanted to make a call and have be burning fuel just to generate the energy to run my phone, right? So we use batteries so that we can more conveniently use our electronic devices anywhere and not have to have them plugged in or not have to carry around a little engine with us everywhere we go. Your cells do the same thing. Your cells will have reactions that occur in very specialized regions in the cell that are optimized to those reactions, so power plants, basically, that will take a fuel source and release the energy. And instead of directly powering an endergonic reaction, or a reaction that requires energy, they're going to transfer that energy to the equivalent of a battery that will store the energy and take it to the reaction that requires energy, and then the battery will release the energy to drive forward that reaction, like muscle contraction or transport or protein synthesis, things like that. The battery in cells is ATP. So ATP, adenosine triphosphate, is cellular energy currency. It is a molecule that is very good at storing energy. So to do work, cells must manage their energy resources by what's called energy coupling, and that's just what I was talking about, is you need to take an exergonic reaction and use that to drive an endergonic one. So like we talked about the other day, you could use that hydroelectric dam to drive a pump to pump water back up to the top again. So that would be an exergonic driving an endergonic one. In cells, most of this energy coupling is mediated by ATP, which, like I said, acts like a battery. There are other molecules. There's GTP, there's NADH, there's NADPH in photosynthetic organisms, there's FADH2. There's a lot of other energy carriers that work in a similar way, but this is the most common one that we see. So if you look at ATP, so it's basically a nucleotide. So this right here would be a nucleotide. You've got a sugar, ribose, you've got a base, and you've got a phosphate. But this one has 3 phosphates. And if you look closely at these phosphates, you'll notice that they have a negatively charged oxygen on one side of them. What do you think happens between those negative charges? Yeah, they are being repelled from each other, right? So opposites will attract, similar charges are going to repel. So these 3 phosphates being together are very unstable. So ATP itself is a very unstable molecule, which means it has, so things that are unstable have lots and lots of free energy. So there are lots of potential energy. If you were to remove one of these phosphates from ATP to make it into ADP, it makes it a much more stable molecule, so it lowers its free energy. And the difference between ATP's energy and ADP's energy is the amount of energy that's released when you remove the phosphate. So this is hydrolysis of ATP releases energy. This energy is what is used then to drive forward reactions. So like a battery, this would be the charged form, and this would be the discharged form. So, and this is the energy used to do cellular work. Okay. So ATP is not a molecule that's used just once in your cells. It can be used many times. If you wanted to go from ADP, diphosphate, to ATP, all you need to do is add in a phosphate and energy to get that to happen. So ATP is renewable. We're going to look in Chapter 6 pretty extensively on this side of this graphic, where you take ADP and how do you generate ATP using energy from food sources, like an energy bar. So ATP is made by the addition of a phosphate group to adenosine diphosphate to make ATP, and then the energy stored in ATP will drive most cellular work. So this ATP can then drive muscle contractions to allow someone to ride a bike, and in the process, the waste product is ADP plus phosphate, so this can be charged up again. So just like your cell phone, you can charge it up at night by plugging it in, so that would be the process of digestion in a cell, and then you have power to play Candy Crush all day. And then when your battery is drained after playing your games all day, you can plug it in again at night and recharge it. So this is what your cells are doing constantly. You are breaking down the food that you eat to charge up ATP. ATP then allows you to move your muscles, and after you've burned through all that ATP, you can charge it up again by eating more food. So we will spend a lot of time talking about this side of things. I'm going to briefly right now talk about this side of things. How does ATP actually drive work? But we're not going to talk about this extensively. You'd learn more about this in kind of physiology classes like that. So ATP, like a cell battery, drives work. And it does it in many ways, but here are just 2 kind of examples of how it might do that. It often will transfer that phosphate, so when ATP becomes ADP, it will actually pass on that phosphate to the molecule it interacts with. And this can then transfer energy to that molecule in one of 2 ways. Adding a phosphate might actually increase the potential energy of the molecule. So if you had, I think I have it here. So let's just say you have a endergonic reaction where your free energy, your reactant is low in free energy, your product is high in free energy. It's possible that adding a phosphate to your reaction or reactant makes it more energetic, pushing your reaction's free energy up above the products, and now you have an exergonic reaction. So you've changed your reactants in a way to make them more energetic, to make them have more potential energy, and so that reaction now will go forward spontaneously and not, it won't require any energy anymore. So you've by adding a phosphate, you've energized that reactant. So that's this first way. The second way, the phosphate could actually physically interact to change the shape of the molecule, and this is what happens when your muscles contract. So if you remember back, we had actin and myosin. Myosin was a motor protein. Myosin has these little heads that attach to actin, and when they become phosphorylated, the heads will bend, and that causes them to pull closer together, and that's how your muscles contract, is by the bending of the heads using the energy of ATP. So ATP bonding actually can change the shape of proteins. So those are just two ways. There's other ways that it might function as well, but ATP can change a reaction to make an exergonic one or endergonic one go forward. Okay, so hydrolysis of ATP takes ATP, converts it to ADP, and releases energy plus phosphate. Right? That is a squeaky marker. Okay. So let's see if you can answer this one. So here's two reactions. Reaction A, carbon dioxide plus water to give you glucose and oxygen, requires energy for that to occur. Reaction B, oxygen plus glucose converted to carbon dioxide and water, releases energy. Which of these two reactions would most likely be coupled with hydrolysis, or the breakdown of ATP to ADP? So which of these two reactions would require ATP to be present? Okay. Please talk with your neighbors and think about this a little more. Do you understand what I'm asking in the question?
You mean which one requires ATP?
Yeah, which one requires ATP hydrolysis? So this one requires energy to go forward. This one releases energy as it goes forward. So which one of these two reactions would this occur at the same time? This one have to happen alongside of it. So coupled means tie together. So which one of these two is tied together with this reaction? All right, so you switched your responses, but still 40% of you are confused. Well, maybe all of you are confused. this is what it says. This is what I have right now. So what I mean by this, one of these reactions can occur without ATP being present. One of them needs ATP to be present in order for it to happen. When ATP is when you break down ATP, what's released? Energy, right? So which one of these will not go forward unless there's energy present? A. Okay, that's what I was asking here. So the answer is A, because A requires energy, so it needs to have this reaction coupled with it. So this will occur at the same time as that reaction, and this will provide the energy for A to go forward. B would do the opposite. B would actually provide energy for ADP to be converted back to ATP. Right? So we're going to see, this is the reaction on the bottom for cellular respiration. So oxygen and glucose, so the glucose you eat and the oxygen you breathe in, is converted into carbon dioxide that you breathe out and water. And in the process releases energy. That energy is captured by ADP, turning it into ATP. Now this one on the top, you don't actually use ATP to power, but you could, and ATP is involved in it. But this is an equation for photosynthesis. Carbon dioxide in water is converted into glucose and oxygen. And really, photosynthesis, instead of using ATP to power it, uses light energy to power it. But it needs energy source to power it. So you could power this with this reaction. So hopefully that makes a little more sense. This isn't a question I'm going to ask you on a test because obviously it was confusing as to what I was asking for. But that requires hydrolysis of ATP, whereas this one would definitely not require it because it's an exergonic. So this would be endergonic, this would be exergonic. Endergonic reactions can be powered by ATP. Okay, that's kind of the take-home message. Okay, so just like running your phone requires energy, and endergonic requires energy. The energy is supplied to your phone by a battery. Energy is supplied to endergonic reactions by ATP. Okay. Okay. The next kind of half of today's lecture is all about something called an enzyme. Okay, reactions don't progress the way I've been showing you. So if we have an exergonic reaction like this, where the reactants have a lot of free energy, the products have a lower free energy, the reaction is not going to progress like that. So the reactants don't just form the product immediately. This would actually be kind of chaotic in cells if every single high energy reactant was immediately converted to a low energy product. So let's go back to an example I used the other day of a diver standing on a diving platform. Diver standing on a diving platform is like your reactant. High free energy, high potential energy. Diver in the water, low free energy, low potential energy. Why does the diver not immediately end up in the water? Okay, so standing on the platform, in order to get in the water, they have to take a step forward or jump forward, right? So that diver, though they have high free energy, is actually locally stable. They can't get into the water unless they put in a little bit of energy to get to a slightly even less stable, higher free energy state of standing over air. Okay, is there anything that's going to stop that diver from ending up in the water once they're standing over air? No. And so that's why you don't see divers just hovering in air for long periods of time. But you do see them standing on the diving platform thinking if this was a good idea to stand up there, right? So standing over air, they'll fall immediately. So what happened in that reaction, rather than going from reactant to product, you went from reactant up to this air, which is your transition state, then straight to a product. So this is actually a little mini endergonic reaction that required energy input, and that energy was the muscle contractions of stepping forward or jumping forward or being pushed by someone forward, right? So once they get here, boom, they go into product. So most reactions will not progress until they reach this transition state. And in a lot of reactions, it's breaking the first bond, it's somehow changing their conformation, it's coming together in the right orientation, it's some change that once it gets to that point, that's the point of no return. This is the point where you can't stop the reaction past this point. Boom, they're going to end up being the product. So this distance between the reactant and the transition state is called the activation energy, or the energy of activation, the EA. So the activation energy. This energy is, in most reactions, typically supplied by heat from the surroundings. So this is why a lot of reactions occur more rapidly when there's heat present. So heat will cause bonds to break, heat will cause molecules to move around. It's going to cause a reaction to go forward more rapidly. So think if you have two reactants that need to come together in the right orientation in order to react, if you heat them up, they're going to move a lot faster and they're going to bump into each other more often and the reaction is going to go forward. If you cool it down, the chances of them actually getting in the right orientation is pretty slim to none. Right? So a little bit of energy is involved in starting a reaction. So let's say I want my diver to end up in the water more quickly. What if I take that diving platform and I cover it in grease? So now that diver standing on the platform, they're no longer going to have to step forward. They might just have to lean a little bit forward and they'll slide right off, get to their transition state, and fall here. So now, instead of requiring the energy of muscle contraction to step forward, they require just a little bit of energy to shift their weight forward, right? So I have lowered the activation energy. So grease, in this example, is what you might call an enzyme. Enzymes are typically proteins, but they're molecules that will speed up chemical reactions by lowering the amount of energy needed to get the reaction started, by lowering the activation energy. So here is a graph where you have activation energy kind of pictured as a barrier here. The reactants are above the products, but they have to go over this big barrier to get started. Activation energy with an enzyme is purple here, it's much lower. Activation energy without an enzyme is the beige here, it's much, much higher. So this process is called catalysis. So an enzyme is often called a catalyst. Catalytic enzyme speeds up a reaction, and it does that by lowering the activation energy. So lowering the energy required to get a reaction to go forward. All right. So, what are enzymes? How do they function? Well, enzymes have different parts to them. Enzymes are typically proteins. An enzyme has a region in the enzyme called the active site. And this is going to play a role as we start talking about how enzymes are controlled. We're going to learn more about what goes on in the active site. Reactants, or the substrate in a reaction, will interact with the enzyme's active site, leading to an enzyme subject complex where a reaction occurs, and then products are released. Key to an enzyme being an enzyme is that enzymes, by definition, do not alter either the reactants or the products. So the products are going to be the same with or without an enzyme. The reactant is going to be the same, or the substrate is going to be the same with or without the reactant. And the enzyme itself is not consumed by a reaction. This enzyme is the same at the start as it is at the end of the reaction. So when we were talking about ATP and ATP being used to get reactions to go forward, ATP is not an enzyme. ATP is changed by the reaction. ATP will actually change the reactant or the product. It actually is physically involved in a reaction. So it does not act as an enzyme. An enzyme is something that just speeds up a reaction without altering what's happening. So my diver still starts on top of the platform, still ends up in the water. They just get there faster, and that grease is not affected by the diver. The grease is still there afterwards. It can now cause the next diver to get in the water faster. So enzymes can be used over and over again to catalyze the same reactions because they're not consumed and they're not changed by the reaction. Okay. So how does an enzyme work? It's kind of the next step to this. All right, there's many different ways that enzymes can lower a reaction's activation energy. Here are a few of them. One is orienting substrates correctly. So again, let's say these are my two reactants. They need to come together in the right orientation. Maybe they need to have their caps bump into each other like this in order to react. One thing that can change the rate of a reaction is heat. So heat is going to speed up these molecules and make them bump into each other faster. Cold temperature is going to make this happen slower. But let's imagine I have an enzyme that's shaped in such a way that when these substrates bump into it, it holds them in the right orientation to get them to come together in the right fashion to react. That enzyme is going to make it easier for the reactants to come together and is going to orient them in the right area, meaning that it takes less energy for them to get into that right formation. They might also strain substrate bonds. So sometimes the first step in a reaction is breaking a bond, and an enzyme could cause the bonds within a substrate to be stressed so that they might start to break more easily. So it's not going to change the substrate in any sense, it's just going to strain the bonds. Providing a favorable microenvironment. So enzymes are proteins, and proteins are made of amino acids. And if you remember back to when we talked about amino acids have lots of different properties. They can be polar, they can be nonpolar, they can be basic, they can be acidic. Let's say that this enzyme, which has maybe a bunch of amino acids here, and let's say this is the active site of this enzyme, and let's say all of these amino acids are acidic. They're going to cause this region within the active site to become acidic as well, which means it has a lot of hydrogen ions. So maybe having all those hydrogen ions is going to put stress on the substrate in some way or change kind of the reactivity of the substrate to make it more likely to react than it would if it was in a more neutral environment. So that's a favorable microenvironment. They could also covalently bond to the substrate. However, unlike ATP, they're not going to stay connected, and the enzyme is going to go back to the way it originally was at the end. So it's not going to ultimately change anything about the substrate or the product, but it could bond to it briefly or in a transient manner to make the reaction happen a little bit more rapidly. Okay, so enzymes speed chemical reactions by lowering activation energy, somehow making a reaction happen at a slightly faster rate. Okay. All right, so what you're going to be doing in your lab, coming up soon probably if you haven't done it already, you're going to study enzymes. Mostly what you're going to be doing is studying the effects of different things on enzymes. And by changing the conditions an enzyme is functioning in, you can change reaction rates, and you're going to be measuring reaction rates. So you're going to be changing the pH, changing the temperature of the enzyme, and then seeing how fast the reaction occurs in those different conditions. So the reason why this is important to understand in biology is that reactions, although they might be exergonic, although they might proceed with a release of energy, most reactions in an organism will not occur, just will not occur at all, without an enzyme present, because this red kind of non-enzyme activation energy level is huge. So activation energy is really, really big. So even though it's a reaction that can occur spontaneously without energy input, it may take 100 years for a reaction or 200 years for a reaction to occur randomly. So without an enzyme, it's basically not going to happen. So reactions will typically only occur inside an organism when an enzyme is present. Because enzymes are typically proteins, anything that affects a protein is going to have an effect on the enzyme. And if you affect an enzyme, you're going to affect an enzymatic reaction. So what we're going to talk about next class are ways that cells or toxins or things can actually interact with an enzyme to change its function or prevent it from working, that can actually allow your body to control which reactions occur and where. But your enzymes are also subject to the environment that they're in. So that involves the pH that they're in, the salt concentration, or the temperature that they're in. If they're not in an ideal temperature, what's going to happen is the hydrogen bonds within the enzyme could break, causing the enzyme to unravel or denature, and then causing it to not function to catalyze a reaction. So this is a graph of rate of reaction versus temperature, and temperature is in Celsius, so you may not completely understand what these numbers mean. But here, a human, most human enzymes function best at 37 degrees Celsius. Anyone know what that might be in Fahrenheit? 98.6, right? So you probably all know that number. What do we call it when your body temperature raises above that number? A fever, right? So if you had a fever, and it's even just anything above that, even 99 is called a mild fever, right? So what if your temperature is like 105? Anyone know what happens? Yeah, you probably die, right? If it's that for a very long time, that's not something that's very survivable. So you'll be dumped in a tub full of ice to try to cool you down, right? So what's happening is your enzymes are all denaturing and no reaction can occur inside of your body. So most organisms function best just below the limit of their enzymes. So because temperature is high, reactions will go forward very rapidly. There's a lot of energy around to get over that activation energy. But if temperatures get too high, your enzymes get destroyed, and all of a sudden your activation energy needed jumps through the roof, so you can no longer get the reaction to go forward. What do you think slows reaction on the other side of things, on the cold side of things? So your enzymes aren't being destroyed by cold temperatures, but why?
Frozen.
All right, well, so what does that mean for a reaction?
They're stopping slower.
All right, so everything's slowing down. So this energy requires heat, typically, to get going, because it requires random movement of molecules, and if you cool things down, those molecules are moving more slowly. So if you look at these graphs, on the left side of this, let's see if I can pull it up so it's not covered by this thing. So the left side, there's this gradual decline in reaction rate, but on the right side, there's a cliff that it falls off. So have you heard of people falling into an icy river and surviving? Right, that happens. It could be deadly, right? A very long time in cold water is not going to be good for you at all. But you can survive for a short period of time being slightly hypothermic. And actually, you can cool your body. Your body can survive even being like 85 degrees. So that's a pretty drastic change in temperature. Have you ever heard of someone falling into a volcano and surviving? Really? Okay, not a... so something that's nonfiction. No. All right. No. All right, so no, because the enzymes all would get destroyed and there'd be no coming back from falling into lava, right? So the upper end of things is not survivable, but the lower end of things might be, because you're not destroying the enzymes, you're just slowing down reactions. Right? Okay, so local conditions will affect enzyme function, and we'll talk a little bit more about these things. You're definitely going to talk about these in your lab. You're going to alter pH, you're going to alter temperature, and most organisms have an ideal state for these to be in. You can draw a similar graph. I don't think I have one on here, but you can draw a similar graph of pH, but it's not as drastic on one side or the other. For pH, you'd have pH, and you'd have a graph that's kind of like this, where you'll have an optimal pH, and things will decline on both sides of those. This actually works to the advantage of your body. You have a very low pH in your stomach, and you have a much higher pH in your intestines. You have certain enzymes that do specific tasks within your stomach, and you don't want them to be functioning in your intestines because you want some other reaction to occur in your intestines. Once you leave your stomach, the pH rises, and those enzymes that were very active in your stomach because of the low pH are going to be inactivated by the higher pH. And just like the enzymes that function in your intestines that function at a higher pH, in your stomach here, they would not function, but in your intestines they would function. So this controls which reactions occur where. You're going to have stomach reactions occurring in your stomach and intestinal reactions occurring in your intestine because pH will control the enzymes in them, right? Sure, it's a control mechanism. So it makes sure that they can only function in the region they're supposed to happen. Yeah, so that's what you meant then, yes. All right. Okay, so let's see if, all right, so here's a graph of a bacteria reaction and over temperature here. Which temperature do you think is denaturing the enzyme? Mm-hmm. So which temperature is denaturing the enzyme? Okay, last few responses, if you haven't already. Okay, so here's what you said. So most of you said 78, so about 68% of you. A few of you said 73, and a few of you said 55. So if I was to draw a line from this peak here down, this is about 73. And here, this is about 78. So what's happening at 78 to the reaction rate? It's plummeting, right? And it plummets because the enzyme is denatured. So the right answer would be 78. What's happening at 55 degrees? So there's slowly declining down that side because of temperature, not because of destroying the enzyme. What temperature do you think this bacteria lives in? All right. Why do you say that? Yeah. So most organisms, their enzymes are ideally suited to function at the temperature that they're found in. So our body temperature is 98.6 because our enzymes function really well at 98.6. Or it might be the other way around. Maybe our enzymes function well because that's our body temperature, and so we've developed enzymes that function that well. So this bacteria, this hot spring bacteria, will live at 73 degrees Celsius. So there are organisms that have different enzymes than us. So even though 55 degrees Celsius would be plenty hot to destroy all of our enzymes, because ours work at 37, this one works best at 73. It's denatured at 78, and at 55, the reaction stops working not because of the enzyme, but due to lack of energy. Okay, we're going to stop here today, and we'll move on to other mechanisms of regulation of enzymes on Friday. Right.